The title of my talk is The Connection Between Alternative Splicing and the Epithelial to Mesenchymal Cell Transition. Don't worry, it's not a cell biology talk, but this process, epithelial to mesenchymal transition, called EMT, is an important process. It's an important plasticity program of the cells, and is active during embryogenesis, during development, during organogenesis, during tissue regeneration, during wound healing, but unfortunately for us, it's also active during metastatization of epithelial tumors, okay, carcinomas, this kind of thing. And I will show you that there is at least two important alternative splicing events which involve two proto-oncogenes that control this phenomenon. And this work was started many years ago in my lab. I initiated it, but in the last years it was carried out by other people because in the meantime I became old and I retired, but I can still tell you about what was going on. It involved this receptor called RON. You might ask, why is it called, does RON means? RON comes from the French récepteur, do you know? Récepteur d'origine nantaise. What does it mean? That was discovered in Nantes? Okay, so it's a receptor, RON. It's a tyrosine kinase receptor for macrophage stimulating protein SFP, MSP, and a member of the scatter factor receptor family. It's a very complicated thing, but it's a receptor. It's a heterodimer made of two subunits, beta and alpha. The cross-membrane subunit is beta. It's a usual receptor, the tyrosine kinase. What does it do? When it binds certain ligands, like the macrophage stimulating proteins and scatter factors, it changes cells. It makes the cells more mobile. The cells, especially epithelial cells, start to move around, to migrate, to cross the barriers. The reason why I became interested in this receptor is because our colleagues of the Cancer Institute in Turin told us that an isoform, a splicing isoform of this receptor was overexpressed in gastric tumors initially, and then it was found overexpressed in many other tumors. And the splicing isoform that was overexpressed was this one. You know, this gene has many isoforms, many alternative splicing. But the particular splicing isoform that was overexpressed was this one that they call Eltiron, and I will call it Eltiron for the rest of this talk. Okay? It has a deletion. There is a small exon, exon 11, that codes for 49 amino acids only, which is skipped in this particular splicing. This small deletion, the deletion of this small exon is very important because what happens is that in this exon, there is the site of recognition of the protease that cleaves the precursor and produces the heterodimer. So in this isoform is not cleaved. So this Eltiron isoform is constitutively expressed, so it phosphorylates the downstream signal cascade, even in the absence of the ligand. So it's hyperactive, doesn't need any ligand to operate. So this is the description of this particular isoform that is kind of worrying, to find it in tumors. It's deleted of 49 amino acids in the beta chain, extracellular domain, and originates from the skipping of exon 11. This exon is skipped. This isoform is constitutively active in the absence of ligand. It confers an invasive phenotype to the recipient cells. So if you transform cells transiently with this isoform, the cells become more invasive. And you can test it with an in vitro assay, a transwell assay in which the cell can migrate through a membrane, nitrocellulose membrane of certain pore size, and you can verify. And this isoform is overexpressed in the majority of colon and breast cancers. So this was the starting point. We did a lot of work to find out what happened, why in certain cells you produce this isoform. This isoform is very bad because by conferring these invasive properties to the cells, they cause the metastatization of tumors. So what causes the skipping of exon 11? We did a lot of work, several papers on them. And we found out that it's kind of odd, Usually what controls the inclusion or the exclusion of an exon is within the exon. In this case, we discovered that what controls the inclusion or exclusion of exon 11 is in the downstream, is an exon 12. The exon 12 has an enhancer of splicing. Professor Barale talked about this enhancer of splicing. In enhancer next to a silencer, but what is important is this enhancer of splicing that contains binding sites, very strong binding sites, for a protein, a splicing factor called SF2-ASF. You have already heard about it. It's an SR protein. It's one of the members of the SR protein. They are splicing factor, essential, involved both in constitutive and alternative splicing. In particular, SF2-ASF is an essential protein. It is a proto-oncogene. When overexpressed in mouse, it causes tumors. Anyway, we discovered that what controls the skipping of exon 11 is the binding of SF2 to the enhancer. And of course, and the binding of SF2 to the enhancer, besides causing skipping of, causes the epithelial to mesenchymal transition. This transition is a change in the cell shape, in the cell organization. So the epithelial cells are they are like cobblestone. They are bound to each other, very tightly bound. Mesenchymal cells are more like fibroblasts. They are elongated sparsely, and so here they are. So they are cells transfected with a clone expressing SF2-ASF. And as you can see, the shape of the cells change, and you have a rearrangement of actin filaments. And then there are several other markers of this transformation from Unfortunately, there is too much light here. You don't see anything. But anyway, there is a strong reduction of E-cadherin. E-cadherin is an adhesive at the junction of the cells. And a reorganization of beta-catenin, which is typical of this transition. E-cadherin here is decreased. This is, sorry, there is a switch. It should be vector here and ASF here. Anyway, and a migration, an increase of the capacity of these cells to migrate through a nitrocellulose membrane. So it's enough to increase the SF2 factor to induce this transition. Not much, twofold, threefold, that's what causes this transition. The transition is reversible. The process is called MET, the opposite process, from mesenchymal to epithelial. And this slide, during this transition, a lot of things happen. You have proteins that increase in abundance, N-cadherin, fibronectin, transcription factors, and so on. Protein that decrease in abundance, E-cadherin, and so on. Protein that accumulates in the nucleus, that normally are in the cytoplasm, they move to the nucleus. And the properties are increased scattering of cells, elongation of cell shapes, and resistance to anoicosis, which is resistant to apoptosis. The cells are more, they don't die easily. So this is the process. The epithelial cells gradually are stationary. Mesenchymal cells are motile and invasive. And of course, this is a model, a cartoon that describes what happens in the carcinoma tumors, especially at the edge of a solid tumors. Some cells undergo this process, and they cross the fibronectin, the barrier that separates tissues, right? Because they can move, and they can even, unfortunately, they can move into the bloodstream and create metastasis in a faraway district. And once they have settled in another district, the process can be reversed, and at the metastasis, usually cells go back to the epithelial form before degenerating again. And so the process is confined to the metastatizing front of the tumor, according to. We discovered that it's very simple to reproduce this phenomenon. It's enough to grow cells. These are colon carcinoma, a line of colon carcinoma cells. If you grow them, you can grow them at high density or a low density. A low density, by simply changing the density of the cells, you switch, and you can see that at low density, the cells look more like mesenchymal cells, while at high density, all the parameters are of those of epithelial. See, for example, here, beta-catenin. Here is in the cytoplasm, at low density, moves to the nucleus. And here they are the other parameters that clearly indicate that simply growing cells at different density, you reproduce what's going on. And, surprising, but not too much, simply growing cells at different density, you change the splicing profile of RON, of the receptor. So at high density, you have full-length RON, so exon 11 is included. At low density, you have almost 90% exclusion, skipping of exon 11. And in parallel, you see an increase of SF2 ASF. So the density of the cells influences this particular alternative splicing pathway. And so we said, well, that's very strange, until a paper was published about 3 or 4 years ago, in which the paper says that also the splicing of SR genes, and particularly of SF2-ASF gene, is controlled in a funny way. Let's concentrate on this one. This is our protein, the protein that, when it's overexpressed, causes the skipping of exon 11 in the receptor RON. This protein has an intron in the 3' untranslated end. And you say, well, who cares? It's untranslated. Doesn't make any difference. But if you splice this intron, you create a junction, exon-intron junction, and you know the story. When this junction is more than 50 or 60 nucleotides downstream from a very good stop codon, this stop codon becomes a premature one. And as you will hear tomorrow from Lynn McQuaid, the DNA, the RNA, the transcript that contains a premature stop codon are degraded, are directed to a pathway of destruction. And this is true for all these SR proteins, these splicing factor regulators. Okay. So this was very interesting because it turns out that this very important splicing factor, this homeostatic level, is controlled by a splicing event in a non-transcribed 3' region. Well, who cares? Until, we observe that the cell density, like it did for the RON receptor splicing, cell density, which is a model of EMT-MET transition, also regulates the splicing, the alternative splicing of SF2-ASF. You see? At low cell density, you have mostly the full-length isoform, so you don't splice this intron. At high cell density, you switch, you produce the NMD plus isoform, and so at low cell density, you have more SF2-ASF, and high cell density you have less. So the transition epithelial to mesenchymal affects in the same way RON and SF2. Now, we did a lot of work I don't have time to go through all of it, but that the phenomenon is real. So cycloheximide and HUPF1 siRNA, so in a silencing of the genes for this is a protein that is involved in the destruction of transcripts that contain a premature stop codon. If you inactivate this protein, in fact, you block the process. But just to tell you that we have all possible controls that actually, indeed, this is what happens. So the splicing of the triprime UTR is altered, not only in vitro in our system of cells growing high, low density, but also in tumors. For example, here you have a sample of 14 tumors, and you see that the ratio between full length and short isoform of the SF2 transcript is altered, In the majority of tumors, you have a lot of full-length mRNA, and so more protein around, because the full length is translated, and we think more protein, more SF2, more Deltaron, more receptor missing the exon 11, and so constitutively expressed. Then we found that not only you can reproduce the phenomenon by growing cells at high density and low density, but turns out that what triggers these changes in the splicing pathway are simply factors which are secreted by cells. You don't need nothing complicated to change the splicing profile of SF2 or of RON. It's enough to use conditioned medium. Conditioned medium, cell conditioned medium, is very complicated. Anyway, you grow cells for 24 hours in a medium that does not contain serum. So the medium does not contain growth factors, albumin, cytokines, all the things that are present in the serum. Then whatever is present in the medium when you remove the cells, it has been secreted by the cells themselves. And so, for example, if you take cells grown at low density and you treat the cells with conditioned medium from cells grow at high sensitive, you see that when you add the conditioned medium, you switch the splicing of SF2 from full length to NMD. So the supernatant of conditioned medium cells makes the cells become like the epithelial cells. So, but these are becoming complicated, and you see here the same thing. Okay, but it's enough to add high conditioned medium to low density cells to elicit all the characteristics of mesenchymal cells. So what triggers this change? What triggers this change are soluble factors, probably, growth factors, cytokines, whatever, secreted by the cells. And usually, these factors act through the mitogen-activated protein kinase MAPK cascade. Through a lot of steps, they start with SARC, and then they go MAP kinase 1, MAP kinase 2, ERK, especially ERK. This is a key transductor of the signal from the growth factors to the nucleus. Okay? So remember this. It's a signal transduction cascade, the main one, MAP kinase. And the secreted factors that control the splicing of the SF2 UTR turns out to be, probably, it acts through the ERK kinase. And this slide shows that the ERK kinase is phosphorylated, very phosphorylated in low cells, very little phosphorylated in high cells, and this is an inhibitor of the phosphorylation of the kinase, and so on. So in any way, the secreted factor controls the splicing through this Ras-Raf-MEK-ERK2 cascade. Believe to my word, there are many other experiments. I cannot show you all the cartoons. Okay, now the funny things happens. What control the splicing of this intron in the 3' non-coding portion of SF2? It turns out, according to the paper that I showed you before, this intron is flanked by two ultra-conserved elements. And when I say ultra-conserved element sequence, it means that there are 200 nucleotides here and 200 nucleotides here that are 100% conserved from mouse to man. And as Lynn said, when something is so conserved, it means that it has a function. But this is impressive, 100% conservation of two sequences in a non-coding sequence. Clearly, this conservation has something to do with the splicing of this intron, which is very important to maintain, to control the level of SF2. What we found in collaboration with the people here in Trieste, Buratti, who talked this morning, Baralle, is that next to this ultra-conserved sequence and the fixed distance from them, there is a binding site for another protein, another RNA binding protein, SAM68. SAM68 is a splicing regulator. It's an important RNA binding protein. It's called SAM68. SAM stands for Sarc activated in mitosis. So it's an RNA binding protein that's been discovered by, I think, by a 2-hybrid system in yeast to be bound to Sarc. Sarc, if you remember, is the first player in the cascade. So this protein is an RNA binding protein that is supposed to be the mediator from the splicing, from the signals of the growth factors down to the RNA metabolism in the nucleus. So there are two binding sites for SAM68. SAM68 is an RNA binding protein. It has binding domains, and it has 3 sites phosphorylated by ERK, the ERK kinase. This slide was done in collaboration with Emanuele Buratti to show in vitro that, in fact, this sequence this is a pull-down experiment made in vitro. In fact, SAM68 binds to the sequence, and here the experiment is done with mutants that they don't bind anymore, and so on. This is an experiment done with mini genes containing SAM68 to show that they behave as expected from the fact that SAM68 has an effect on the splicing of this intron. In other words, when SAM68 binds to these two binding sites, the intron is not spliced. Okay? And SAM68, in doing so, controls the production of these transcripts which are degraded by the NMD. You see, if you overexpress SAM68, for example, in high cells, in low cells, you expect to see cobblestone-like appearance. Instead, SAM68, by including this exon, it increases the level of SF2. You increase SF2, you increase deltaron, and you have fibroblast-like cells. It's very confusing, but to simplify things, I made a cartoon. This said the same thing, that SAM68 controls the production of NMD, also of the endogenous SF2 transcript, because the previous experiment was done on mini genes and so on. But in the end, and this slide shows that as SAM68 mRNA expression level in human is altered in human normal colon and tumor tissues. So whenever you have high level of SF2, you also have high level of SAM68. So there is a correlation between SF2 and SAM68. So our data suggests that SAM68 expression level or its phosphorylation status could play a role in tumorigenesis. So I don't want to bore you. I prepared for you a cartoon that summarizes, here it is. It's a very nice summary of what's going on. So post-transcriptional regulation of epithelial to mesenchymal transition. Our idea is that in low-density cells, for example, and also at the leading edge of tumor that begin to metastatize, what happens is that some signal, some secreted factor, phosphorylates RAS, then phosphorylates RAF, then MEK, and then ERK, and so eventually it phosphorylates SAM68. And we have the evidence that SAM68 is phosphorylated. Phosphorylated SAM68, or overexpressed SAM68, protects this region and avoids this splicing, which is deleterious because this splicing leads to destruction. So in this way, you have an excess of SF2, which impacts on the splicing of deltaron, and you produce deltaron and you activate the empty probe. So you see, you have two splicing regulators, SF2 and SAM68, that act on each other in a very delicate balance to control eventually the splicing of another protein, of another gene, which is a protein that directly controls probably the plasticity of cells. Is it clear? So, do I have 5 minutes more? Let me check. I was supposed to end 15 minutes ago, but we are 15 minutes late. No, I wanted to show you. So these observations gave us and to other people the idea that it is possible to correct the splicing of deltaron and use these drugs or whatever they are as anti-metastatic therapeutic targets. And I just describe you 3 approaches that have been used in our lab in collaboration with other people. One approach is this one. If you block the enhancer where SF2 binds with morpholino antisense oligonucleotides. Morpholino antisense are oligonucleotides which are modified into sugar so that when they hybridize to the RNA, they don't cause degradation of the hybrid by the RNase H. So when you transfect with this morpholino, in fact, you see a certain reduction of the effect. So you include exon 11. The problem with this approach, I don't have time to go into it, but the problem with this approach is not shown here, but is not shown here, but these morpholino derivatives inhibit the splicing of intron 12, of the other intron. So it is specific, but it has this side effect. Another approach is this one, is TOES, targeted oligonucleotide enhancer sequences. So this is an oligonucleotide, modified, of course, to avoid this degradation of RNA, which hybridizes to a sequence of exon 11, specific, therefore, for exon 11. It has a tail that recruits splicing factors like SF2 and so on. And in fact, this approach is more promising. You see, the transfection with this oligonucleotide reverses in a cell line that has prevalently delta RON, you go back to RON. And this is specific. So this is to show that it has no effect on another caspase gene. These are caspase gene, and this is another gene which has an SF2 target site. Doesn't do anything. It's specific for this gene. And it doesn't do anything on the splicing of exon 12. So this is more promising approach. TOES oligonucleotides. So they bring to the exon you want to preserve the splicing factors that are required. And lastly, there is an approach where people, in collaboration with a French guy called Jamal Todd, University of Montpellier, this guy has screened a lot of indole derivatives, small molecules, pyrrolizole, I don't remember the names. Some of these indole derivatives, act on SF2, so they inhibit the phosphorylation of SF2, and therefore the binding and the activity of SF2. Some of them are promising. In particular, one, this one, you see, is the one that reverts more. And of course, the small molecule-based therapies are easier because, you know, the problem with the oligotherapy is that it's difficult to deliver the oligos into the cells, and not to say about the organism, while small molecules could work. But anyway, just to mention that there are approaches that eventually will lead to experimentation to reverse this splicing of RON into delta RON. That is probably not the only event, but certainly an important event in going from a tumor cells to a metastatic tumor cells, mobile tumor cells. Okay, thank you. I hope I conveyed the message.